Our next speaker is Dr. Agata Exner. Dr. Exner is professor in radiology at Case School of Medicine. She is also a co-leader for the cancer imaging program at Case Comprehensive Cancer Center. So as you can see, UH is metastasizing quickly across the Comprehensive Cancer Center leadership realm. Agata is an internationally renowned researcher leading multidisciplinary efforts on the development of nanobubble contrast agents for ultrasound molecular imaging, as well as engineering novel nanomedicine and long-acting injectable formulations for ultrasound-enhanced drug discovery. Today, she is going to speak on personalized nanomedicine using contrast-enhanced ultrasound and predicting efficacy of nanomedicine-based chemo.
Good morning, everybody. Thank you for inviting me to give this talk. I am really excited to share with you our work on personalized nanomedicine using contrast-enhanced ultrasound. So our work really lies, as Quentin said, at the interface of nanomedicine, biomedical ultrasound, biomedical engineering. We do work in the preclinical and translational space, and hopefully in a few years, in a short few years, we'll be doing clinical trials as well with this technology. So I just want to take a step back and give you some context. So we're really interested in our group with our collaborators to pushing the boundaries of ultrasound imaging. Ultrasound, most people think of as a modality that's been around for a really long time. There's been no innovation. You image babies with it, that's great, but what else can it do? And so here's just a snippet of everything that ultrasound can do and is doing currently right now. So the technology is old, but it's really undergone a revolution and an exponential growth in the technology in the last decade, I would say, and not the least of which is POCUS. You may have heard this term, so point of care ultrasound. Miniaturizing those big clunky ultrasound scanners that you're used to seeing into a single transducer, which is wireless and it can run through your cell phone. And so really this is democratizing biomedical imaging diagnosis and image-guided therapeutics and putting an ultrasound in every physician's pocket. So it's really a tremendously capable technology. As you can see here, some other examples of super resolution ultrasound. So you think of it as some grainy old images, but what you can really do is get sub-micron almost resolution. This is kidney vasculature. You can do functional imaging in the brain. You can detect tumors with really high fidelity. So there's a lot that it can do. It can replace all of the costly scanners that you're used to seeing these kinds of images with. One reason for why ultrasound is so interesting is the contrast agents that it uses. So typically you think of an iodine contrast agent, maybe a radioisotope, a nuclear imaging. So in ultrasound, the contrast agents, which are FDA approved already, used primarily in echoes in cardiology, are particles. They're microparticles, they're bubbles actually. So these are gas-filled little lipid balloons which circulate, stay in the blood pool, and it can give you really beautiful edge definition, vascular definition, as you see here, an example in a heart and then in a liver tumor. There's been no innovation in this field in 20 years or more. Actually, these agents were developed some time ago. And so there's really ripe opportunity to take advantage of this very unique platform. What my lab has done is really try to miniaturize this platform into a nanoparticle. So why do we want to do this? There's a good reason for that, and that is that it enables extravascular application. So all of the interesting stuff in terms of therapy, in terms of molecular imaging, target detection, specific cancer biomarkers, not all, but most, I would argue, lies extravascular in the tumor microenvironment. So yes, you can target tumor microvasculature, and people have done that for decades. But if you can get beyond that and you can exploit the really great technology that's in hardware and software that's already available with ultrasound, we can do a lot more. And so we've shrunken the microbubble to the nanoscale size, and I'm going to show you one particular application of doing that. And so ultrasound with contrast enables images like this. It's real time. It's completely quantitative. You can follow the progress of a single microparticle, nanoparticle in a tumor blood vessel that creates the opportunity to have very unique biomarkers. You can look at these. These are called time intensity curves. And with that time intensity curve, you can track at 20 frames per second or faster how the signal, how the contrast agent moves through a region of interest. You can do this in 3 dimensions. So this, I'm showing you 2D planes, but there's technology already existing where you can look at the entire tumor volume and gather this information. So that really creates a unique set of potential biomarkers and information that's accessible to physicians already. That we can use to do a variety of things. So some of you may be familiar with our work with Jim Basilian, where we are creating nanobubbles that are targeted to PSMA for prostate cancer imaging and detection. So this is the world's first cancer cell targeted ultrasound agent that we've created. And the idea is to go back to transrectal ultrasound and use the contrast agent in conjunction with transrectal ultrasound to do prostate biopsies. Right now, you may be familiar with, and the buzzword in prostate ultrasound is MRI. There's MR detection, MR-guided biopsies. But most of the urologists, most clinics that do not have access to MRI would prefer to stick with transrectal ultrasound. So we've created this construct, and we're really trying to push this towards commercialization. We're in large animal trials right now, but hopefully you can appreciate that this is just an example of an orthotopic tumor. This isn't a rabbit model that we created, where in the prostate with ultrasound, in the prostate with MR, you cannot really pick up a tumor, but this is exactly the location of the tumor. These are the exact planes where this tumor should be. And so if you squint your eyes, you can see it on a regular truss. But with the nanobubble enhanced data, you can really create very beautiful maps with targets that pinpoint where the tumor location is. So this is work that we've been pushing for over a decade. We're in the canine model now, and we're looking for investors to get us through the valley of death because we've really come as far as we can with this technology. So if you're interested, come talk to me afterwards. But I want to take a step back into a new application that we're looking at, and that is predicting the efficacy of nano-based chemotherapy. So most of you are aware that nanomedicine has sort of overpromised and underdelivered in the past couple of decades. There have been a ton of particles that have been developed to package anti-cancer drugs. Many of them, probably 90% of them, fail in clinical trials. This is just an example from a paper that came out last year showing that there are 45 different nanoparticle constructs being tested in 80 clinical trials. This year, that updated number is about 114 clinical trials. A good amount of them are in cancer. 9 out of 23 nanoparticles that are FDA approved right now are for cancer therapy. But again, there's a ton of potential. You can use it for diagnostics, for theranostics. Right now with immunotherapy, there's a resurgence in interest in packaging things, as well as with gene therapy into nanoparticles. But the challenge is, again, the tumor heterogeneity. So a lot of the thinking behind why nanomedicine has not really become as useful as it could is because of the heterogeneous tumor microenvironment. We heard a little bit about this already. Tumors can be necrotic. Some of the vasculature in tumors can be leaky. Some of it may not be leaky. It's just a big mess. And it's very difficult to predict which tumor in a patient has the phenotype, has the leaky vasculature, maybe has a less dense microenvironment that will allow a nanoparticle to get to it, to come out of the blood vessel, to target and get into a specific cancer cell. So this has been a huge hurdle in the field. As a result, there has been a lot of interest in trying to predict and develop methods that can help stratify patients for the enhanced permeability and retention effect, or EPR. And so there have been a number of different assays. This has been implemented clinically, and they range from a simple blood test all the way to imaging-based strategies. And I really like this graph because blood tests are very simple to implement, but they're very nonspecific for this particular question. Histology and IHC is somewhere in the middle. But really, if you can develop an imaging technique that can predict this non-invasively and accurately, you can get incredible specificity, and then you can help determine which patients will be responsive and which not. And so the question that we've asked in the lab, of course, as you may imagine, is if nanobubbles combined with ultrasound can answer this question. And so this is, again, in a preclinical stage right now, but we think that with some adaptation of existing tools, this can become a reality clinically. And so this is good because we could develop a tool now that can determine how leaky, let's just say, tumors are, non-invasively using a very simple modality that can be placed in an oncologist's office even. It can also serve as a tool to further study tumors very easily, look at longitudinal studies in tumors, look at how tumor progress and how their leakiness changes so that we can deepen our understanding of these processes. And so I just wanted to show you results of a preclinical study. This study is being led by Michaela Cooley, who is an MD PhD student in my lab. So we try to test this hypothesis by comparing this nanobubble-enhanced ultrasound movement and biomarkers in leaky versus non-leaky tumors. So she found there's a ton of literature on this kind of stuff. It's going down 30 years. And there are some tumors that are very well described and characterized and known to exhibit a high EPR phenotype or a low one. So this is two examples of that. Glioblastomas, the U87 glioblastoma specifically, are known to have very low permeability versus the LS174 tumors, which is a colorectal adenocarcinoma. It's known to be very, very leaky and permeable. And so let me just go back here. She did a study where she injected our nanobubble contrast agents. She then determined from those images that I showed you these time intensity curves, and then we try to find which parameters from those time intensity curves correlated to a doxil uptake in the tumors that was done as a terminal experiment right before the mice were euthanized. And so I'm just going to show you some examples of what we learned. So if you look at, let's see here, I'll use my pointer. We looked at the rising area under the curve, so the area under here until the peak from the start of the signal. We looked at peak intensity, and we looked at a decorrelation time. We looked at a number of others, but these are the ones that seem to correlate really well. But let me first backtrack here. When you look at the rising area under the curve, so this is how quickly the signal reaches peak intensity inside of the tumor, we saw big differences, but we only saw them when the tumors were larger. So when the tumors are smaller, and here the blue tumors are the leaky ones, and then the red tumors, hang on a second, this is wrong. The blue tumors are the non-leaky ones, the U87s, the gray ones are the leaky ones. So when the tumors are developing, when they're small, when they're sort of in the micrometastasis stage, you can imagine none of them are very leaky, it turns out. So they were very equivalent. But when they start to progress and grow, the vasculature becomes more disorganized and chaotic, and they become much more permeable. And then we started to see very large differences between the leaky tumors and the non-leaky. The physiology behind these biomarkers is still a bit of a mystery to us, and why it takes longer to reach peak intensity when the tumors are leaky. I mean, we can come up with some explanations, but we're not sure. The same thing with peak intensity. So there's no difference when the tumors are smaller, but there starts to be a difference when the tumors progress. Peak intensity was a little bit complicated, as you may imagine, by the tumor heterogeneity. So if you just look at two examples of these maps, these are tumors that showed a very high peak intensity, but in some regions, and these were low peak intensity in most regions. But you can see it's complicated by necrotic areas, it's complicated by regions that don't get a lot of contrast. And so this is the same problems that we're encountering with the nanobubbles that are encountered with nanoparticles of any sort. Some tumors just get a lot of them, and some don't, and that leads to incomplete and insufficient treatment. And so we decided to stratify these tumors into quadrants instead of looking at average areas of interest. And then if you compare these biomarkers in each quadrant, so just take, for example, one of them. If it has a high area under the curve, it correlates with high signal intensity from the drug. And you can see that being manifested here. We also developed another image processing technique. This was developed by a student, Dana, in my lab, and it's an autocorrelation map. So we're looking at decorrelation time mapping. The idea here is that it's essentially measuring the rate of change of a signal in a particular pixel or voxel. So if you think of bubbles that can come out of the vasculature and get lodged into tumors, attached to tumor cells, their signal eventually over time isn't going to change a lot. And so this is what we're picking up here versus tumors that just have blood vessels that are non-leaky where the stuff is moving very quickly. And so we can develop from this a leakiness index. And that seems to correlate extremely well with the amount of doxorubicin that's found in all of these different areas. So you can see, again, we can predict with very high fidelity here which areas of the tumor and which cross-sections will have a lot of drug and which not. So what we don't know yet, and these studies are ongoing now, is does this prediction and uptake translate to greater therapeutic efficacy. Those studies are going on right now, and so I'll be able to tell you about those later, but not today, unfortunately. There's a lot that we can do with bubbles. I showed you a little bit about this idea of companion particles in nanomedicine. I think this approach can really have a significant impact. Big pharma could be interested in this approach because it can lead to them having better outcomes in their clinical trials and just a deeper understanding of the tumor models that they're using. So I think there's a ton of potential here. There are a lot of different applications of bubbles, one of which that we're pushing and I don't have time to talk to you about right now is a therapeutic application. So because these are bubbles, they're gas-filled vesicles, we can actually collapse them with an ultrasound pulse completely non-invasively. And that bubble collapse, when the bubbles are at their target, leads to very high apoptosis in tumors and a decrease in progression of the tumor. So I don't have the data here, but I wanted to tempt you, and I'm happy to talk to you about it later on. So thank you again for the invitation. I'd like to acknowledge my lab, all our collaborators, and I'll take any questions. You probably shouldn't have done that. Hang on.
Let me start off then, Kata. So, as you pointed out in your talk, tumor vasculature is often very disorganized and chaotic. And, there's a school of thought that these leaky vessels actually enhances drug penetration in the tumor. But there's also an opposing view that because they're so leaky, you can't cover the entire tumor. Have you given any thought on using your platform to sort of measure response to chemotherapy in patients?
Yeah, that's a great question, and we have. I think you can definitely do that. So there are a couple different applications. One, you can simply look at how the signal distributes in those tumors as a potential response. So the same physiology that's driving nanoparticles out of the tumors, like high interstitial pressure, for example, should have the same effect on the nanobubbles floating through the tumor, so we should be able to pick that up on images. The other very interesting thing, there's a technique that's being developed, for example, to use these bubbles to measure interstitial pressure. And so clinically, there are clinical trials now looking at how the bubbles change their signal based on the ambient pressure around them. So the idea is that if you have a tumor that's responding to chemotherapy, the interstitial pressure will actually decrease instead of increasing. And so that can be used, and there's a clinical trial right now at Thomas Jefferson and maybe some other sites that's looking at this as a potential marker of prediction of chemotherapy success. So there's a lot of really interesting applications. Thanks.
In your, that was really wonderful. In your image where you showed the targeting of the microbubble using the prostate targeting agent, it was, really quite impressive.
Thank you.
Obviously, can that same tool be used not only to image, but then to deliver targeted therapy?
Yes, exactly. I think that's something that we're really interested about. So some of you may have heard of high-intensity focused ultrasound that requires these really massive pieces of equipment. It's a wonderful technology and it works really well. But if you can focus the ultrasound and the energy just with targeting the bubble, then you can use something that's very inexpensive. So in the lab, we use physical therapy ultrasound units to pop these bubbles. These units are a couple $1,000 and you can buy them pretty much anywhere. We buy ours on eBay. And so that removes the necessity to have the costly equipment to drive the therapy. And you can have the therapy be driven by the bubbles collapsing by themselves, or you can load the bubbles with drug. People also use bubbles to sensitize tumors to chemotherapy, to radiation therapy. There's clinical trials out there looking at that. So absolutely, we're focusing it in a different way, and that's definitely something we're investigating. Thank you.